So thanks for coming along. I think I know most of you. So I'm going to kind of cover what we do in the biology theme. I guess everyone will know something, but hopefully not everybody knows everything. So I'm going to kind of cover the broad sweep of what we do in computational biology, and going through a few exemplar projects that we have done and that we are doing. Then at the end, I've just got a couple of slides on future directions, hoping that might spark a little bit of discussion. So just to do the history bit, to say where we came from. Back in the day, we were a small CCP4 team based at Daresbury, supporting the SRS as was. So we were actually not a team in scientific computing or CSE. We were actually a team in the synchrotron radiation department. And those are our origins. That's where we're coming from. Around 2005, I can't remember exactly, we started moving into molecular dynamics simulations to complement the crystallography that CCP4 was doing. And then in 2009, the CCP4 team moved down here, first into Diamond House. So I think about 6 months or so, we were based in that big open plan office on the right of Diamond House until they finished building the research complex, apart from the leaky roof. And then we moved into the research complex, which is where the CCP4 and the CCP-EM teams are now. I should say the molecular dynamics activity continued up at Daresbury and is still there. CCP-EM was created in 2012, looking at biological cryogenic electron microscopy. That's very much following the model of CCP4. That started in 2012 and is still going strong. In 2020, computed tomography group led by Eduardo Pasca was added to my group. And then in January of this year, as part of a reorganization of our department, the computational biology theme was created, which basically covered these four activities. So just to summarize what the computational biology theme is now, we have the macromolecular crystallography group, which is basically the CCP4 team, biomolecular simulation, which has grown out of the molecular dynamics activity, molecular and cellular electron microscopy, which is the CCP-EM team, and then computed tomography and imaging, which is Eduardo's team. So formally, the computational biology theme is covering these four areas. However, the idea of the themes within SCD is that they're not just organizational units. They also cover activities across the department. So anything remotely biological in the departments, then I can claim it. So in practice, obviously we talk to other themes, other groups within the department. The materials theme does a lot of molecular and mesoscale modeling, and we work with them where appropriate. Engineering group would normally be a bit away from biology, but they do a lot of finite element modeling. And it turns out that for the larger biological structures, finite element modeling is useful, and we have collaboration going with those on mesh generation. We talked to the maths group over various algorithms, SciML for machine learning, DARS, including IDARS for providing services, and a lot of our own services are backed by SCD Cloud. So for this Ada Lovelace talk, I'm focusing mainly on the big facilities here on this campus and how we've interacted with those facilities. But I mean, I should make clear that we all have two lives, as well as kind of focusing on the facilities here. We also a part of national and global communities. So in practice, a lot of those have run through the CCPs. So these are the collaborative computational projects. These are national consortia, but they have plenty of international links and generally would operate on a global scale. So when we're doing our stuff, writing software, writing services, we have one eye on the facilities here and one eye on the external community. Of course, in many cases, we share users. So a user would come to Diamond to collect data and then go away and and become part of the national community and use our software there. So that there is clearly overlap. But when we're writing stuff and designing stuff, we have an eye on things that would be applicable to the wider community as well as bespoke solutions for facilities. So just to kind of end this first section, to summarize our interests, and computational biology is a bit of an exaggeration. I feel honor bound to, whenever I'm talking at STFC, to remind STFC people that biology is a huge area. Lots of stuff that we don't do, lots of biochemistry, lots of imaging, lots of organ modeling, population modeling, ecology, etc. Loads of biology out there that we don't do. We largely focus on molecular and structural biology, but kind of expanding out a bit in the imaging area and expanding out a bit in the bioinformatics area. But I guess at the end of the day, that's our core business. Because of our background, our origins in SRD, synchrotron radiation department, we've always been closely tied to the experiment and to the generation of experimental data. We do modeling and simulation. And I'll come on to details later, but we do modeling, but it's largely with the aim of interpreting experimental data. I think we would rarely do modeling in isolation. So where we are now, we've grown organically over 30 or so years. I would like to claim that there was a plan, but there probably wasn't. But we have grown. I'm going to put numbers up. There are about 35 staff in the computational biology team now, plus associated hangers-on. So we're now quite a large activity that's grown up, as I said, and have a variety of funding. Historically, we've largely had external funding from research council grants or charity grants or European grants. And our core aim then is really the development of software, methods and software. Through this kind of longevity, we hope that we provide a stable home for software. When we're talking to university partners, the contrast is always with the postdoc who writes something and leaves. The idea is that we're the postdoc that stays. So we have lots of experience and expertise in writing software and maintaining it over years. We do develop methods, although I would say they tend to be fairly practical methods of how to deal with problems. We're not really hardcore theorists, I would say. There are exceptions to all of this, but in general. And again, linking back to experiments, I think we've always had a close eye on data and metadata handling, because that's crucial for getting the most out of experiments. So that's all a bit of blah, blah. What does that mean? In practice, what that means is that we're developing software libraries and frameworks and workflows that support various experiment-based methods. I did a matrix diagram because I think it's kind of obligatory for this kind of talk. I'm not sure that it tells you very much. Down the left-hand side, the kind of operations that we do. So we're dealing with experimental data. doing modeling and simulation, handling data and metadata, and then there's a ubiquitous machine learning. And along the top, there are roughly the kind of experimental techniques that we would be concerned with. So diffraction experiments, microscopy, tomography, and then scattering, and thinking about reflectometry and small angle scattering and things like that. And yeah, there's a nice matrix there. We have probably projects in each element. So with that kind of introduction and preamble, I'm now going to quickly survey a few projects that we have done or we are doing. So kind of starting with our core business, which is computational structural biology. These are the CSP4 and CSP-EM projects. Now, I think a lot of you in the audience will know these, but I'll just go back to basics for those that don't. So CSP4 is a long-running project and software suite. We claim the origins are 1979, but no one really knows for certain. That software suite that starts from diffraction images and processes the data, solves the phase problem, builds an atomic model, which can then be deposited in the protein data bank. CSP4 now has a large software suite that has grown up over many years. This is downloadable to your home lab and has a variety of user interfaces. That software is there on the Diamond MX beamlines for general use. Also, bits of it are incorporated into the automated data processing that the Diamond does. And Diamond have also done Diamond also have their own processing pipelines. So it's which kind of under the hood use some elements of our software and other elements. CSP-EM was created later, 2012, which was 2 years before the resolution revolution officially took off and cryo-EM became trendy. But it was set up in 2012 largely to mimic CSP4. So again, for the cryo-EM, we started with single particle analysis, and that's going from the frames collected at the microscope through to a final map and potentially a built atomic model. Again, we have a software suite which you can download and run locally, and that's available at EBIC, so Diamond's electron microscopy facility. And that's available to use as is, and it's also underpins some of the pipelines, which I will mention later. So for CSP4, one thing to highlight is CSP4 Cloud. So this is a cloud-based service, which is mainly run here, backed by STFC Cloud. And users can log in, have access to most of the CSP4 suite, upload their data, and do all of the structure solution in the cloud. That's been running for a few years and is getting increasingly popular. An issue has always been that it's quite problematic to upload the original diffraction images to CSP4 Cloud. So people tend to come in a bit later. They do the initial processing locally and then upload the reduced data and work with that. So Jules, who's in the audience, I'm probably going to, I'll kind of name a few people as I go along. But Jules has been working on a data link to help the transfer of large data sets, particularly fraction data sets, to and from the CSP4 cloud service. And there's an API set up now, which will allow you to push data to CSP4 cloud. You can also pull from the cloud if you're logged in. And there's a Globus endpoint just recently been set up. So the infrastructure is there now for easier transfer of data to and from CSP4 cloud. And they're just kind of working with diamond at the moment to try and smooth that. So this is general technology which would allow anyone to work with imaging data onto the cloud, but it is specifically aimed at diamond and the MX beamlines, so that there will be a seamless transfer from data on the diamond data stores up into CSP4 cloud, where you can continue the processing. More generally for diffraction, CSP4 kind of grew up with X-ray diffraction, but of course there's increasingly electron diffraction, I can't remember what we're allowed to call it these days, not micro-ED any longer. Electron diffraction is increasingly popular in the bio world, and neutron diffraction as well. So each of these diffraction techniques have a very similar workflow, but of course there are differences. So for electron diffraction, you have a very flat eval sphere, which messes up some of the standard data processing. For neutron diffraction, you have to process the time of flight data. So within like the CSP4 family, and in collaboration with Diamond and Lawrence Berkeley, we have the DIALS software suite for doing that initial data processing. So this is processing diffraction images and taking you as far as integrated Bragg peaks. Then it can go into the rest of the CSP4 suite. So DIALS was conceived many years ago, 10 years ago, more, okay, 12, 15 years ago, as a replacement for a CSP4 program, which was getting long in the tooth. So it was conceived as a kind of new next generation software suite for X-ray diffraction processing. But since then, it has been adapted for electron diffraction and neutron diffraction. So what we have now is a DIALS suite with a core library, a core set of operations, which is general, but with particular customizations depending on particle type, source type. So as an example of that, David McDonough has been working on neutron dials for neutron diffraction. And here the issues are the time of flight, so the multiple wavelengths that you have to put into the data processing, unusual peak profiles, and the detector geometry that they have at ISIS. So these are all kind of customizations that have gone into the DIALS suite, and actually that led to some tidying up of the code, which has then had impacts for the rest of the DIALS family. But particularly the dealing with the time of flight setup at ISIS means that the wavelength is explicitly there in the data processing, and now that DIALS is suitable for X-ray Lowry diffraction, and there's now been interest from Harvard group to do X-ray Lowry diffraction on the back of work that was done for ISIS. For electron diffraction, Marco has been working with the EBIT guys on a workflow for electron diffraction. Issues here are that the micrographs or diffraction images are written out with metadata, but the metadata's split over several files. Some things like the beam center are usually missing. So Marco has written an auto-ED program that runs at EBIC, which will detect when images are being written. It will analyze them, automatically determine the beam center, and then check for various other pathologies, and then write out the corrected and tidied metadata into a Nexus file. And then that's all set and can be sent off to the diamond cluster for processing using CO2 and dials. So this has been done as a kind of step on the way to automation. So previously automation efforts have failed because of incomplete metadata or kind of poor management of data. So this project has specifically addressed that so that we can now do automated processing at EBIC. And that was, I have to say, that was done quite quickly. I think that took about 6 months to get all of that sorted out. So moving into the EM world. So I mentioned the CCP EM software suite. The bit for this talk is the pipeliner, which is the business logic layer underneath the CCP EM suite. So this has been written from scratch in the last few years. And it's based around an acyclic graph data flow. So all tasks are given input and output nodes, and these can be joined together to give you a graph of the processing that you're doing, and particular attention done to kind of metadata collection and capture and recording. So this is kind of the business end of CCP EM. Within Pipeliner, you can also define user interfaces, and then these are then realized in the Doppio interface that we have. It's designed as an open architecture, a plugin architecture, so that it's relatively simple to put extra job types into it. So this is work of Matt in our group with Shaw's and Gerard in Cambridge. Sorry, Gerard, I should say, wrong one, Gerard. But the point of having this kind of logic layer underneath all of this is it can then be used in different ways, not just to support the GUI. So it has been used at EBIC to define automated processing for single particle analysis. So again, so this is distinct. I was just talking about electron diffraction. So I'm now talking about electron microscopy, single particle analysis. So this is a different use case, different technique. So EBIC have a workflow that runs whenever micrographs are collected, and that's written on top of the Pipeliner architecture. That's available for users to run as well, but it's also used with 1 or 2 customizations by EBIC. So that was done with Dan Hatton at Diamond. Yeah, and the end point of that is some processed or semi-processed data which goes into iSpy-B. That goes in with all of the project information that Pipeliner has collected. So then it's possible for the user to go to iSpy-B and download the endpoint of this processing as a CCP EM project. And then that can be imported directly into CCP EM and taken further down the line. And the other point of capturing all of this metadata is so that you can archive projects later. So this is true for the EBIC end of it. It's true for later downstream as well. So at any point in this workflow that Pipeliner is recording, you can click on a job and say, please tell me how I got here. What is the data flow that got me here? What were the jobs that I ran that got me here? And that can be archived either as a full archive with all of the files, or as a script which you can then go away and run again. And the ultimate goal of all of this is that if you can move from EBIC to initial processing to CCP EM and keep all of the metadata and all of the workflow, then you should be able to deposit that to the international archives. And we do have a job now which will package up files, including original micrographs, movies, and send it off to Empire. So Empire is an international repository for electron microscopy raw data, and they have a script which you can use to automatically upload. So we collect all of the metadata, we give it to the script, and it can be uploaded to Empire. And we're now talking to Kyle Morris, who used to be at Diamond, now at the EBI, about working on something similar for PDB and EMDB endpoints. So I've done electron diffraction, single particle analysis, and the other major technique is electron tomography, and using improvements in the Reliant software, which have come through recently, and the pipeline architecture. We've created the first basic pipeline for electron tomography and subtomogram averaging, so that you can then begin to automate tomography pipelines. Tomography is a bit more difficult. There are a variety of programs, and people tend to chop and change a little bit. So it still needs work to make it a completely general solution. But proof of principle of doing a tomography project through a pipeline is there. And in general, for the electron tomography, we're kind of working very closely with people at UBIC and RFI, and also externally at Cambridge and Crick and other places. So yeah, Rangana's in my group, and he's the one responsible for this. So moving on to modeling and simulation. I would have to say that we've done modeling for a long time. I mean, some people forget that when you build a crystallographic model, something that you deposit in the protein data bank, that model is not an experimental observable. That is a model of your diffraction data. So that's a bugbear, because people tend to download from the protein data bank thinking that's an experimental result. So we've been doing modeling for a long time. That often involves getting structural models from a variety of sources, whether it's other experiments or from homology modeling, these days from AlphaFold, and tweaking them to fit the current data set. Traditionally, that's what's done in molecular replacement, and we've had pipelines for that for a long time. But it's now also done in cryo-EM in trying to generate models to fit into the map. And the name of the game here is really, identifying, if you model a structure, being able to identify which are the good bits and which are the bad bits. And you're often going through the process of trimming down the model to the high confidence regions so that you get the best agreements with experiments. And even with the magic of AlphaFold, well, I don't know about AlphaFold 3, that might be completely magic, but with AlphaFold 2, certainly it gives you kind of good regions and bad regions, and you do need to go through a process of quality control and looking at the core of the structure, which is reliable, before reusing that model. And so that's the kind of thing we've been doing in CSP4 and CSP-EM for a while now. But I won't talk about that anymore. Talking more about molecular simulation. So for us, that principally means classical molecular dynamics, and that's taking a structure from wherever and seeing how it evolves in time to get some ensemble information or some dynamical information. So a few use cases. So we've been working with RL and Luke at ISIS on neutron reflectometry. They're looking at these complicated layer structures, typically kind of lipids or other surfactants monolayers with embedded peptides, proteins, glycans, whatever. And neutron reflectometry gives you a good Z profile through your your sample. Traditionally, that was modeled with a simple kind of continuum layer model until people realized that you could get that you could fit atomic models to that data and get a little bit more detail out of the experiment. In a sense, the atomic models are higher resolution than the technique, but I think you're using the facts that you're bringing prior knowledge of what, chemical knowledge to play into this. So you can use molecular dynamics to produce an ensemble of structures and then look to fit that to the neutron reflectometry curve. So we've tried a few things: bilayers, monolayers at the air-water interface, and self-assembled monolayers on a gold substrate. And the coolest thing that they probably do is these floating bilayers. So in the top right here, the top bit of the sandwich is a bilayer, then you have a water layer, then you have a SAM layer on top of gold. And they've shown that you can tune how the bilayer floats above, depending on salt concentration and other things. So those are the kind of things that we're wanting to model. One thing we realized is that the fit to data is very, very sensitive to how you set up the simulation. One thing you don't really know is the area per lipid of your layers. And rather than guessing, we've adopted a technique of systematically fitting different simulations to the data. And you can use the fit to the data to tell you what your best area per lipid is, and hence what your best model is. So this is a good case where we're not just doing the molecular dynamics in isolation, but we're looking very carefully at the fit to the data, and we're using the experimental data to feed back to the simulation. Feedback to the simulation. In this context, I want to mention ShapeSpire as well, which is doing very similar things for small angle scattering. This is not formally within the biology theme, but it is partly biology. So ShapeSpire is toolkits that Andre Bruckner in a different theme has been writing. So this will take a lot of the steps that you require to set up a molecular dynamics simulation and generating different starting structures, different aggregates, setting up and running an MD simulation, and then doing various analyses afterwards. So it's a very nice environment for kind of having control of a simulation and outputting things which are then useful for comparing to the small angle scattering data. And one of the reasons that I brought this up is that we're now trying to join together the reflectometry project with the small angle scattering project, because they have very similar concerns and aims. So Valeria, who works with me, is now working with Andre, putting layer geometries into ShapeSpire, putting NAMD workflows in where there was GROMACS before. Another technique that we've just recently made contact with is QENS, quasi-elastic neutron scattering. So working with Mona Sata at ISIS, these kind of based on the time resolution that you can achieve in the neutron scattering, you can get out different timescales of motions that are happening in a protein or protein complex system. And from that, they're able to estimate. So we're looking at a particular system, a streptavidin biotin system, and looking at the binding, and particularly looking at the entropic contribution to that binding free energy. So the entropic contribution comes from changes in the conformational states that you have, and also from displaced waters. So the name of the game here is to try and separate out the different contributions to see what's driving the binding. So Mona's group can get out estimates of entropic changes from the from the Quens spectra. And for comparison, we can do this from an MD simulation as well. So we run an MD simulation of the system, and then within CCP Biosim, we have a new code entropy, which will look at the trajectory from the MD and then estimate the entropic contribution, and then in particular split it over different, down to residue level or looking at the waters or whatever. So these are just results hot off the press where we're trying to compare the experiment and the simulation. There is some discrepancy at the moment, and we're trying to work out why that is, and maybe that reflects some changes of different thinking or different definitions. And the fourth technique that I wanted to mention is going back to cryo-EM. One thing that single particle analysis can do, because you're looking at individual proteins, you in principle have a snapshot of all possible conformations of the protein on your EM grid. Traditionally, what is done is to classify those into discrete classes. Recently, the people are working on more continuous variation in the structures. So heterogeneous reconstruction algorithms, things like cryo-dragon that some of you may know. So cryo-EM in principle gives you a snapshot of the motions of a protein. Obviously, MD is also trying to do the same thing. So what we'd like to do is to compare the flexibility of a protein as seen by MD with that as seen by cryo-EM. They're not necessarily the same because there are different samples, different conditions, but they ought to overlap at least a little bit. So towards this aim, we're kind of working on some benchmarking so that we can begin to understand the experimental heterogeneous reconstruction algorithms. And this means working again with synthetic data, but here we use molecular dynamics to set up a range of conformational states that we know, and then using parakeet platform developed at RFI, we can generate synthetic electron micrographs, and then we can go through the whole cycle of doing heterogeneous reconstruction and identifying either discrete classes or a latent space. And then because we've set it up in the first place, we can then compare that back to ground truth. So in this fourth panel here, there's a little example. This is a latent space of conformations that has been derived from the experimental data. You can then cross-correlate that with what you had in the MD simulation. And you can basically see that the MD simulation carves a path through latent space. So there's a kind of kinetic connection between all these conformations, which is then revealed in the cryo-EM data. So I just wanted to wrap up the MD bit of it with, try and bring everything together, but don't look too closely at the details. This is just a mind map. But the idea is that we've got a variety of experimental techniques, reflectometry, small angle scattering, cryo-EM, Quens, that are using MD to generate models or give you some idea of the static structures or the dynamics of the system. And these all have associated software for fitting the experimental data, but using MD information as an input. On the other side, there's a whole set of, there's far more software than this being written for setting up and running MD simulations. So in an ideal world, it would be good if we, our theme, ALC, whatever, if we could bring all of this together in some way and have some kind of platform that sets up and runs MD simulations, but is also aware of the experimental tests that are going to come afterwards. And that's particularly useful, and something that we've investigated in reflectometry is feedback, so that the fit to the experimental data is not the end point, but you use it to go back and inform the simulation. So if you have this kind of feedback loop, then having some kind of architecture that ties it all together would be very useful. I think at the moment we have a bit of a mishmash, is probably the truth. Right, I am going to run out of time, I'm sorry. Try and go a bit faster. So I've mentioned at the beginning that, yeah, data and metadata management is very important. I think all I wanted to say here is that we're not the guys that do the data storage or the data transfer. There are other people in SCD whose job it is to do that. Our job is really more from a pipeline and workflow point of view, is to keep track of where everything is, and trying to keep metadata and not lose metadata. And one reason for being interested in metadata, of course, is machine learning. So it's the bane of anyone that's trying to do machine learning, is to try and get some decent training data. Even if you can find training data, it's usually not annotated very well. So ends up that there's not a lot that you can do with it. So we have, over recent years, we have done several machine learning projects, and I'll give a couple in a moment. But we're doing it from a very practical point of view. We're not in the business of inventing new architectures or anything, but we're looking at how kind of established machine learning architectures can be applied to our kind of data. And in this, we've worked closely with the SciML group in our department, with the RFI as well, I should say, and also Alan Lowe's group at the Alan Turing. So just a couple of examples of that. So Affinity VAE by Yola and collaborators, many collaborators, I should say, is looking for the identification of particles in cryo-electron tomography. So if you collect these tomograms of real systems, you end up with quite a complicated collection of particles of objects. And you essentially want to cluster them so that you can tell, you know, I've got a bunch of ribosomes here, I've got a bunch of proteasomes here, whatever. So it comes down to like a clustering problem with very, very noisy data. So they developed this Affinity VAE. The autoencoder does the basic job of mapping the particles onto a latent space and then decoding them back again. But the problem is that the latent space is dominated by different views of the same particle. It's very hard to disentangle rotational and translational changes from actually different molecules. Hence the affinity regularizer comes in, which looks at how similar molecules are. So it basically tries to arrange latent space so that similar molecules are next to each other in latent space, irrespective of their rotation and translation. And at the bottom, there's just an example of how that's been done. The bottom left is a representation of the latent space, and it's showing how particles are clustered. The right-hand side is the what they correspond to in reality, and you can see that similar particles go together. I am going to have to speed up slightly. Denoising is very useful for noisy data, which especially what you get in electron microscopy. Noise-to-noise algorithms are very useful because you don't need clean data for training purposes. So Ding has come up with this, or adapted this noise-to-noise algorithm for denoising electron tomography. I'll skip quickly over that. Cryo-DAN is another one looking at 2D particles, and it addresses the problem of when in cryo-EM, lots and lots of particles. Some are good, some are bad. You do lots of classification to try and get rid of the bad particles. This is a more attempting to be a little bit more sophisticated than classification, to try and learn what are good particles and what are bad particles. Hence, clean your particle data set, and hence get better reconstructions. I wanted to quickly cover computed tomography. This is Eduardo Pasca's group. A lot of it is materials, but they also work with CCB Cineby, which is medical imaging. So just a couple of examples. So working with the IMAT instrument at ISIS, they've looked at particular reconstructions of time-of-flight neutron tomography. And in particular, one thing that Ado's team is very good at is looking at appropriate regularizers. So regularizers take features of the data and smooth them in some way so that you don't get so that it's well behaved. So the bottom or the right-hand side kind of shows basically the effect of a regularizer. So this is an energy spectrum that you get from the time of flight experiment with traditional filtered back projection. It's all over the place. Put in a decent regularizer, then you get much smoother data, and in particular you can then identify the Bragg edges, which tell you something about the crystalline phase. Digital volume correlation is where you're looking at a series of tomograms, very often a time series, and you're looking at deformations and going along that time series. We've worked with Ryan Bay at Oregon on his software, but looking at porting of code, parallelization, and GUI. Ado's group have also looked at electron tomography, and in medical imaging, various projects looking at PET and MRI imaging. I need to skip over a little bit of this. So I should say, I mentioned that the IMAT work is obviously with ISIS. The DVC work has largely been with diamond beamlines, I13. And we're also now working with EPAC in the preparation for the tomography that they will do there. Since they're starting from scratch, we've been able to get in there and start building workflows for them and a GUI for the users. Digital twinning. I mentioned briefly Parakeet before, which is for digital twin of an electron tomography experiment, and we expanded that to single particle. And Frank within Ado's team has been doing a digital twin for computed tomography. Right, can I just, yeah, sorry, I skipped over that stuff, but obviously we're happy to talk afterwards about any of these projects in any of the details. But this is my penultimate slide where I just wanted to say a couple of things about future engagement. So we're doing a lot of different things. So now I'm talking general for the computational biology theme, not necessarily for Ada Lovelace, but we have this, I mentioned at the beginning, we have this organic growth, and we would like to continue this organic growth. We kind of stay true to our particular strengths, but also expanding into new areas. We have a lot of experience with software engineering skills, but we are not an RSE group, and we're not. I wouldn't portray us as coders for hire. We do software development for specific scientific domains, and we try to understand the scientific domain. We have been using machine learning, and that's certainly something that we want to build on and do more of. There's lots of machine learning, which is basically object recognition or classification or denoising. I think there's also a potential for looking at the data processing that's being done at the facilities and trying to learn, you know, what's a good experimental setup. Can we kind of do data collection in a more intelligent way rather than the what the crystallographers used to call the American method, the shoot first and ask questions later? Can we kind of go ahead and use machine learning to learn what works and use that to inform future experiments? I guess our main output is automation, it's software pipelines. However, I think, we do like talking to end users. It's very useful for us to make sure that our software actually works with real data and to learn the requirements of the user community. So I think we're certainly keen to engage with end users as well as facility staff. Themes for the future. Software kind of across the facilities. So I've kind of mentioned examples of dials and very briefly maybe I didn't mention it, ADO's SIL package, which kind of bring together common elements across the facilities. Doing more MD for interpretation, imaging, going across more length scales and metadata. So I think I better finish there. And if you want to follow up on any of those things, please come talk to us. We try to be friendly. So that's me and my henchman. If you'd like to come and see us. Okay, thanks for your attention. And I'm sorry for